Okay. 32. Okay. Good morning, everyone. My pleasure today to present Benjamin Dehay from the Institute of Neurodegenerative Diseases in Bordeaux. He's an INSERM principal investigator and at the EMN, which is a renowned research center focused on brain disease. Ben did his PhD at the laboratory of Anna Bertolotti at the École Normale Supérieure in Paris, where he worked on Huntington's disease with an axis in protein aggregation and cell biology. He then got a Marie Curie fellowship and he moved to the laboratory of Miguel Vila in Barcelona, where he focused on Parkinson's disease and studied the implications of lysosomal impairment and dysregulation of autophagy. He finally moved for a second postdoc to the laboratory of Erwan Bessard in Bordeaux with a Marie Curie reintegration grant. But he soon got his permanent position as INSERM researcher, and this is where we met. I arrived to Bordeaux when he just had gotten this position. And we started working together in mouse models of PD, particularly the inoculation of Lewy body fractions from PD patients, which I'm sure he will explain briefly during his presentation. In fact, Ben is involved in a lot of projects about PD, involving both in vitro or in vivo models in rodents, but today he will talk about two recent papers performed in monkeys, which I believe is a model that recapitulates better the clinical aspects of Parkinson's disease. So, okay, I'll leave you to it, Ben. Welcome to Achucarro seminars.
Okay, so hello everybody. So first of all, I would like to thank Federico for the invitation and also for giving me the opportunity to present the new data regarding the primate-specific susceptibility to neurodegeneration and synucleinopathy induced by patient-derived alpha-synuclein extracts. So, as you may know, Parkinson's disease is characterized by the loss of dopaminergic neurons in the substantia nigra pars compacta, which lead to the destruction of the nigrostriatal pathway and then leads to motor behavioral impairments such as rigidity, akinesia, postural instability, and tremor. So to date, the current treatments are only pharmacological and surgery, and they only are based for symptomatic treatments. There is no neuroprotection treatment or neurorestorative treatment for Parkinson's disease. So Parkinson's disease is characterized by the presence of protein aggregates. So in the case of PD, we have the presence of Lewy bodies, which are shown on the top of this slide. And for other synucleinopathies, you can see the presence of glial cytoplasmic inclusions, or GCIs, which are present in oligodendrocytes. And alpha-synuclein is the most protein component in both protein aggregates, the Lewy bodies and the GCIs. We know that alpha-synuclein undergoes an aggregation pathway with different steps over time. So at the beginning, it is an unfolded monomer or either a membrane-bound folded monomer. And then there is a changes of conformation, which leads to the development of pathological conformers. And then we will have the presence of oligomers, protofibrils, fibrils, and finally the Lewy bodies over time during the course of the disease. So this is a hot topic in the field of Parkinson's disease because so far we don't know for sure the pathological conformers and the toxic species of alpha-synuclein involved in the pathology. So alpha-synuclein aggregates can corrupt many cellular pathways in neurons, such as it can interact with the ER-Golgi pathway, the mitochondrion, the UPS, the autophagy lysosomal pathway, the plasma membrane, and also at the synaptic level with the vesicle recycling process. So alpha-synuclein can cause many cellular defects and then which all of them will cause the death of the dopaminergic neurons. In 2008, there is a new hypothesis with the spreading, the prion-like hypothesis of the spreading of alpha-synuclein. It was described by Brack and colleagues that alpha-synuclein can spread into the brain, starting to the medulla oblongata and then goes to the midbrain and finally to the cortical areas. So the big question is how to model disease progression in animal models, for instance. So there is different ways to do that. So we can use age-related models, such as spontaneous model, but there is no ecological models or natural models of Parkinson's disease in all the species studied. After there is the neurotoxin model, so there is different neurotoxins available in mice, rats, and monkeys. And after there is also the genetic approaches, either with the development of transgenic animal models or also to use viral vector mediated gene delivery with all the mutations that is known in Parkinson's disease to help to understand the pathology. And the most recently is the use of inoculation of pathogenic disease-related protein, and here with the inoculation of alpha-synuclein aggregates. So based on the last point, so there is different ways. So either we can use alpha-synuclein recombinant species. So the most known is PFF, which means preformed fibrils, which was firstly developed by Virginia Lee in the United States and Kelvin Luke. Also, there is a possibility to use pure recombinant species, such as the fibrils and the ribbons developed by Ronald Melke's laboratory. And the other approach is to use directly brain from PD patients, so either total homogenates, and our approach is to characterize and to purify Lewy body aggregates. So basically, there is a huge difference between the two classes of alpha-synuclein aggregates after inoculation. So we can use either microgram of PFF to induce pathology, or with the use of brain, we can use a tiny amount to induce pathology around picogram of alpha-synuclein per microliter. So I will explain how we can purify the Lewy body. So first of all, we extract the Lewy body and purify in a second step through a gradient sucrose at different layers, and we can find the Lewy bodies at the interface between the 1.4 molar and 2.2 molar sucrose. We validate the Lewy body presence by different assays, and here is shown the colocalization with the thioflavin S, which is a marker of the beta sheet structure. Then we perform in vitro toxicity assessment, and finally in vivo toxicity assessment in mice, and we can obtain after 4 months of injection, only cell death in the Lewy body injected animals. So this is the pilot study published in 2014. And in this study, we went a step further as a proof of concept, and we performed some injection in monkeys. So the first monkey was injected in the striatum, and the other one was injected into the nigra. So we had the opportunity with José Obeso to follow the dopaminergic innervation through PET imaging. And so each animal follow every 3 months a PET imaging to follow the neurodegeneration. And we can observe that after 9 months post-injection, we can detect dopaminergic denervation, which was confirmed at 12 months. And after, we perform a post-mortem analysis, which also confirms the dopaminergic cell death at the level of the striatum and at the level of the substantia nigra pars compacta. So then we wanted to go a step further, and we launched a large-scale study with a huge number of monkeys for two reasons. First, to reproduce our own results with more animals, and also to ask several questions. So today, because of time, I will present two studies based on this experimental design. So the first one is a comparison between two different alpha-synuclein aggregates, so the Lewy body, and the no Lewy body, which is other aggregates of alpha-synuclein, which was not toxic in mice. And we wanted to achieve an exploratory approach to understand the mechanism of neurodegeneration behind the inoculation and the mechanism of cell death. So first, we use the same paradigm. So we extract and purify the two species, so the no Lewy bodies and the Lewy body. Here it's a dot blot assay to validate the presence of alpha-synuclein aggregates. We tested our fractions in dopaminergic primary culture, and we observe dopaminergic cell death after treatment of Lewy body and no Lewy body in those cells. And we tested also in mice to confirm the toxicity of the Lewy body before to inject them into monkeys. And so in the no Lewy body, we have no toxicity in mice, and in the Lewy body, we have around 40 to 50% of dopaminergic cell death in mice. So then we inject the Lewy body and the no Lewy body, and here is presented the results after 2 years post-injection. And as you can see, at the level of the striatum and at the level of substantia nigra pars compacta, we observe neurodegeneration in both groups at the same level. So around 40–50% of cell death at the level of the nigra, 25% at the level of the putamen, and also 40% at the caudates. So it was completely unexpected to observe dopaminergic neurodegeneration with the no Lewy body, which it was expected to get neurodegeneration with the Lewy body, but the no Lewy body was completely a surprise for us. So we wanted and we need to understand the mechanism behind the toxicity between the no Lewy body and the Lewy body. So to do that, we use a new approach with the selection of a cluster of parameters. So in the first cluster, so we wanted to address the neurodegeneration aspect. In the second cluster, we wanted to address if there is some behavioral impairment. In the third cluster, we wanted to assess the protein aggregation aspect. Here in the fourth cluster, it was all the inflammation, the proteostasis impairment, so the proteasome and the autophagy pathway, and also heavy metals analysis, because it has been shown that it is involved in Parkinson's disease pathology. And in the fifth cluster, we wanted to also detect if there is some occurrence of alpha-synuclein changes in biological fluids. So we choose a different color code to follow the different variables. And we select 40 brain regions in the brain. And at the end of this analysis, we obtain 180 variables measured in all the monkeys in this project. So as you can imagine, we face a question regarding how we can analyze this huge amount of data. And after discussion, we decided to move with a machine learning methodology, which is called the perceptron. And we wanted to ask two questions. Can we identify a minimal data set to diagnose our animals? And the second question was, can we identify the strongest predictors to predict the neurodegeneration? So, we use machine learning methodologies. So in the input layer, we put the 180 variables with a combination of 3 by 3 each time. And in the output layer, we ask to the computer to predict the neurodegeneration. So in the first, we give the raw data for the dopaminergic neurodegeneration in the substantia nigra, here in the caudate, and here in the putamen. So there is a network learning step, and after we obtain the selection of the best learners to predict the dopaminergic neurodegeneration. And here are the results that we obtain to the Lewy body. So this is the kind of pattern that we can obtain. So we can follow with the different color code based on the methodology used, and you can select the strongest predictor, so the first one, to the less powerful predictor. And this is like a signature of the pathology in the Lewy body induced monkeys. So basically, we obtain expected variables, which makes sense in the literature, but interestingly, we obtain also unexpected predictors among all the variables that we analyzed. And this is something new to use like multidisciplinary approaches to unravel novel PD-related pathways. So here is the two signatures, one on the top for the Lewy body injected monkeys, and on the bottom for the no Lewy body injected monkeys. So as you can see, the pattern is completely different if you look at the color code, and the strongest predictors are not the same for the Lewy body and the no Lewy body. So we decided to make a selection and to select only the 12 most powerful predictors to analyze. So for the Lewy body signature, which was interesting, most of the variables are related to the striatum and to the cortical areas, and mainly related to aggregation pathway. So the first one is, for example, the level of phosphorylated alpha-synuclein in the entorhinal cortex. Here in the parahippocampal cortex, we have the level of IBA1 in the putamen, the level of the GABA in the GPI, etc., and etc. So the first variable was a good answer because it was the most highly enriched variable, and it was also a confirmation of the recurrence paper in 2014 in another species, and we observed the same thing in 2014. And for the no Lewy body signature, the first one is the level of the zinc in the substantia nigra pars compacta, and also variables related to aggregation pathway. So after, with all this data, we wanted to make some association between the variables and the place into the brain. So we use another methodology, which is called the LIFT. It's a machine learning approach. So I will explain how it works. So the larger and wider the circle, the higher the frequency, and the thicker the line, the stronger the association in this map, in this ball of wool, we can say. So I will present just two examples of association. So here we have an association between a behavioral variable, which is the actimetry, with the level of phosphorylated alpha-synuclein, which is a marker of the pathological alpha-synuclein in the sensory-motor cortex. So these two variables are clearly associated, and can be and make sense. It is really mainly related with a behavioral parameter. Here you have another example with an aggregation pathway. So here we have the level of phosphorylated synuclein in the VTA. Here the level of alpha-synuclein in the substantia nigra pars compacta. And there is a different association with the putamen, the caudal part of the putamen, with the synuclein, which also makes sense into the nigrostriatal pathway. So there is a link between alpha-synuclein aggregates between the substantia nigra and the putamen. So here is the signature of the no-Lewy body injected monkeys, and as you can see, it's completely different. There is no overlap between the two signatures. And here again, some example. Here we have an example of autophagy connection between the level of the LAMP2, so it's a marker of the lysosome in the putamen, and the number of autophagosomes in the putamen, which makes sense because the two autophagosomes and lysosomes are connected into the autophagy lysosomal pathway. Here it's a connection at the level of the GPI and the CODE, and the level of the GABA, which also correlates, associate with the synuclein in the caudate. And here also it's a strong association between the level of the monomeric form of alpha-synuclein and the presence of high molecular weight of alpha-synuclein in the same structure of the putamen, caudal part of the putamen. So we have with the data, we can see that there is a decrease of the monomeric form of alpha-synuclein and an increase of the high molecular weight. So there is a shift in the balance of alpha-synuclein. So after these results, we wonder what are inside of those fractions in the no-Lewy body and the Lewy body. So we perform a different biochemical assay to understand the content of those aggregates. So we first analyze by thioflavin S, which is the presence of beta sheet structure. So we can see that both no-Lewy body and Lewy body are positive for the thioflavin S staining. We perform also electron microscopy to see what are the shapes of aggregates, and we observe that the no-Lewy body are mainly related to small oligomers compared to Lewy body, which are more fibrillar structure of aggregates. And also we perform different assays, so either proteinase K treatment, which is an assay which helps us to understand the resistance of degradation. And as we can see, we can observe that the Lewy body are much more resistant to PK compared to the no-Lewy body. And we perform also the same assay with the different detergents, the UE and the SDS, and we observe the same aspect. So the Lewy body are really strong aggregates, and the no-Lewy body are much more labile compared to the Lewy body. And finally, we use another alpha-synuclein aggregation assay to show the distinction between the solidity of the aggregates. We also perform a gradient, which also allow us to distinguish between the different kind of aggregates, between small aggregates, intermediate aggregates, and large aggregates. And also with an equilibrium gradient, we can also distinguish between the different kind of aggregates. So basically, what we have observed that in no-Lewy body, we have around 10% of aggregated form of alpha-synuclein, and for the Lewy body, we have an enrichment of 90% form of aggregates. So the content of aggregates is completely different between the no-Lewy body and the Lewy body. So the first take-home message of this talk is the first idea is we are able to follow and to model the prodromal phase of neurodegenerative changes are occurred, because in the Lewy body injected monkeys and no Lewy body injected monkeys, after 2 years post-injection, we can start to detect small behavioral impairments, but we are still below the threshold of symptoms activation, because we have only 40 to 45% of cell death, and we need 50% of dopaminergic neurodegeneration to really obtain a motor impairment. So this is also the first study using machine learning methodologies to establish through an unbiased approach disease-specific features of Parkinson's disease pathology. And the most interesting thing, conclusion is maybe we face a species effect, because if you remember correctly, when we inject no Lewy body in mice, we have no toxicity. And when we inject no Lewy body in monkeys, we have toxicity. So maybe if we use mice or rats, maybe we can miss some key information if we focus on rodents, and maybe there is some toxic species which are not toxic in mice, but can be toxic in monkeys, and maybe also for the human synergy, it is something that we have to think. Another conclusion is the toxicity of the alpha-synuclein, which is contained in the brain of the PD patient, compared to the synthetic alpha-synuclein, where we have to inject a lot to observe phosphorylation of alpha-synuclein and also neurodegeneration. And also the final conclusion that we can think, it's not easy to model PD, because here we can see that we can cause Parkinson's disease, or at least Parkinson's disease phenotype, by injecting 2 different alpha-synuclein aggregates, and the signatures are completely different. So we can model different form of Parkinson's disease depending of what we use to model PD. And we know that in PD patients there is different form of Parkinson's disease. So it's really not be easy to model one form of Parkinson's disease, and when we need to explore many ways to model the disease. So the next step is to use this approach to see how with a treatment, if we have to test, I don't know, a gene therapy, cell therapy, pharmacological treatment, is to use this approach and to see how this treatment can modify the pathological signature, and to see what are the best treatment to change and to reestablish a clear environment and healthy environment. So we want to link a molecular view of this machine learning methodology to move to therapeutic-based approach to understand the pathology. The next step also is to make the same kind of approach with the 6 months and 2 months, 12 months injected monkeys, to see if there is some differences in the signature which are obtained after 2 years post-injection. So this is something that is ongoing. So also I want to present a follow-up of this big project, which is based of again another hypothesis which have been developed by the BRAC colleagues. So they suggested that maybe there is a gut-brain axis in Parkinson's disease, and Parkinson's disease can start into the gut, because we know that there is some non-motor symptom such as constipation, which also can be a sign of the beginning of Parkinson's disease much more earlier than in the brain. So to try to give some clues and to validate or not validate this approach, we use our model. So we inject Lewy body into the striatum, and we inject Lewy body into the gut. And we let those animals during 2 years before to perform post-mortem analysis, as we did at the level on the brain and also in the enteric nervous system. So here are the results. So first I will present the results that we have obtained in the brain. So regarding the Lewy body injected into the striatum, so we obtain neurodegeneration in the striatum and in the substantia pars compacta. And surprisingly, the Lewy body which were injected into the gut, we observe also dopaminergic neurodegeneration, which have been quantified here. And interestingly, we can see that we have the same level of dopaminergic neurodegeneration in the substantia pars compacta with 40% of cell death, and also into the striatum, into the putamen, and much more into the caudate. So this is completely unexpected to obtain dopaminergic neurodegeneration after injection into the gut. So we characterize the synucleinopathy in different brain regions. So at the level of alpha-synuclein or phosphorylated form of alpha-synuclein. And as you can see, the pattern is completely different between the monkeys injected into the striatum and also injected into the gut. So the route is completely different to induce synuclein aggregation. So then after those results, we wanted to see if there is some enteric nervous system pathology. So we analyzed the gut of those animals, and we perform different immunolabeling to see if there is some changes in alpha-synuclein expression. And at our great surprise, we can see that there is some alpha-synuclein pathology in the monkeys injected into the striatum, and also expected somehow in the Lewy body injected into the gut. We have a huge increase, almost the same increase, and we can establish a correlation between the level of the TH into the putamen and the level of alpha-synuclein into the neuron-specific enolase, which are neurons which are presented to the gut with the intensity of alpha-synuclein. So then we wanted to see if there is some aggregation in the gut of those monkeys, so either in the antrum or in the fundus of those monkeys. And we are able to detect some changes in the alpha-synuclein expression, and also with oligomeric form of alpha-synuclein with the use of different antibody. And here is presented the SYN-01, which is the antibody which recognizes the oligomeric form of alpha-synuclein. And then we wanted to understand what are the route of propagation between the gut and into the brain. So the famous hypothesis is the vagus nerve. So we dissect the vagal nerve, and we perform different immunolabeling of alpha-synuclein, phosphorylated alpha-synuclein, and so on in the fibers and also into the cells of the vagus nerve. And we were not able to observe any changes of alpha-synuclein or pathological alpha-synuclein in the vagus nerve of both animals, either the one injected into the striatum or into the gut. So then we analyze the biological fluids of those monkeys, and we observe a huge amount of alpha-synuclein in monkeys injected in the brain or in the gut, which were correlated with the level of alpha-synuclein into the neurons present into the duodenum of those animals. which can be also another route, a systemic route of alpha-synuclein transport between the gut and into the brain, and the brain to the gut. So the conclusion is we were able to observe a bidirectional route of propagation between the gut to the brain and the brain to the gut. And we don't know, we were not able to observe any changes into the vagal nerve and also the biological fluid, and mainly the whole blood. So this was the first study to show that there is a two-direction routes between the brain and the gut. So how alpha-synuclein can travel is still open. So in our case, we cannot observe any vagal nerve alteration. Also, this work raised a question regarding the enteric nervous system biopsy, which is sometimes used in Parkinson's disease patients to confirm the Lewy pathology. So maybe we can detect alpha-synuclein pathology, but this is not a marker to say that this is the start of the pathology, because maybe the pathology starts into the brain and maybe the gut is concomitant to the brain pathology. So this is why the question of the enteric nervous system biopsy as a biomarker for the human patients. And also the big question, we are not able to answer the question of the chicken and the egg, because we don't know if the pathology starts into the brain or into the gut based on the results, as there is a connection between the brain and the gut in those monkeys. So maybe we have to assess, to redo the experiment, maybe to make a kinetics, maybe at 6 months, 12 months post-injection, and to see where the pathology starts first between the two organs. So this was a huge study during 7 years. So many teams were involved, the team in Bordeaux, the team in Barcelona with Miquel Vila, the team of José Obeso in Madrid, the team of Maria Trinidad Herrero at Murcia University, and also Nantes laboratory for the gut approach, and many other collaborators to analyze the different biological fluids and also different protein aggregation assays. So I would like to thank for your attention, and I will be pleased to answer to all your questions if you may have. Thank you.
Thank you, Ben, for a fantastic talk. Okay, you can let me know in the chat if you want to make some questions. Otherwise, I have some questions to make on my own. Okay, so I think I will start with one where we. Okay. Amanda, she has a question. I will close my microphone so she can ask. Amanda, let's go.
Maybe you can write.
No, it's okay. She doesn't. I can. Yeah.
Okay.
You can write it down. Let me see.
So it's a good question. So I think I don't think that it would be the case, because we know that there is a lot of bacteria into the gut, and even there is no proof that by eating I don't know, some contaminated alpha-synuclein or contaminated foods, it can be have some problem, at least for synuclein. For the prion, it's completely another story. But for alpha-synuclein, we know that alpha-synuclein aggregates are not so much resistant compared to the prion protein, for instance, the prion scrappy. So I think we cannot get sick by eating some alpha-synuclein, because I think we need also much more content and quantity to get sick. And after, it's not related to prionic, because in the human pathology, we can detect alpha-synuclein aggregate into the gut, but also in SC patients. So these things, some things that may be physiological, that maybe we can, with aging, we can have alpha-synuclein aggregates into the gut, but this will not cause Parkinson's disease. Maybe it's a problem of proteostasis. There is a problem of clearance, and maybe some proteins are sensitive to clearance impairments, and maybe some of them can be aggregated. But this is not the reason that may be toxic. So the question is still open, but I think we have to first well characterize the alpha-synuclein aggregates and to determine the alpha-synuclein species which are toxic. So I think we are safe so far.
Okay. Fabio?
Hi, Ben.
Hi.
So thank you very much. Impressive work. So I was thinking, so you showed that no LB fraction also has some few aggregated forms of alpha-synuclein. So did you speculate a dose-dependent response to different concentration of aggregated alpha-synuclein? I mean, can you speculate that few aggregated alpha-synuclein can have a different effect on neuronal death or neuronal biology respect to a high quantity of aggregated synuclein?
Yes, because based on our results, we can see that no-Lewy body are mainly oligomeric forms. And we know that oligomeric forms can make much more much more perturbations at the cellular levels. So they are much more easy to make some holes into the mitochondria, the lysosomes, to block the traffic of the vesicles and the synaptic vesicles. So depending of the content and the quantity of alpha-synuclein oligomers, maybe no-Lewy body can be much more toxic compared to Lewy body, which are much more bigger and can be much more inert compared to the no-Lewy body. So now we try to dissect and to analyze with atomic force microscopy the no-Lewy body content, and to see if there is some, I don't know, 1 or 2 or 5 different oligomers, or it's maybe only 1 or 2 isoforms of alpha-synuclein. So this is something that we have to assess. But so far in our hands, the no-Lewy body in mice, even if there is some oligomeric forms inside, the mouse cannot, the mouse is not sick with the no-Lewy body injection. So maybe there is a species barrier between the oligomeric, between the human and the mouse alpha-synuclein. So this is something, another question that we have to answer. And maybe it's mainly related to human or monkeys connection. So maybe it will be interesting to understand the content of the no-Lewy body.
Thank you, Ben.
Okay, I have a couple of questions, actually. I find it very interesting that the non-Lewy bodies the no-Lewy body fraction triggers the generation in monkeys, because you know, we work in mice and it never happened in mice.
Yeah.
So I think it's interesting because it's more representative of what might happen in the patient. I mean, I don't think in the patient there are, I mean, the Lewy body they form, but I think they form after many years. In the beginning, you should have something resembling the non-Lewy body.
For sure.
And the signature from the perceptron that you get from the non-Lewy body monkeys is interesting, first for the zinc, which was unexpected. So I wanted to ask you if you have looked more into the zinc thing. I don't know if you check on patients or the CSF of patients, or if you have some perspective to do something about the zinc, because it's the best predictor for the non-Lewy body.
Yeah, so we are currently analyzing the zinc. So we perform different experiments with a PhD student. So we wanted to see the toxicity of the zinc by itself. So we perform Lewy body and no Lewy body injection in mice, and we perform different treatments with zinc. And we use a chelator of zinc, which is called the cleokinol, which is able to chelate and to decrease the content of zinc. And as we have seen, that if we treat mice injected with Lewy body and no Lewy body, we can have protection if we decrease the level of zinc into the substance nigra and into after systemic injection of cleokinol. So I think there is a clearly strong correlation between the level of zinc and the neurodegeneration, because a lot of people work on iron and also manganism, but there is few studies involving the zinc. And the zinc is important at the lysosomal level, at the ER stress level. It's important for the cellular pathway and for the DNA replication. And so we are currently analyzing the contribution of the zinc in the pathology. So this is something ongoing.
Okay, the second question is more like an insight on, I keep comparing the results in mouse and the result you got in monkeys. First, the autophagy, because one of the image that you show for the perceptron, you show that in the non-Lewy body you have some results from autophagy markers like LT3 and LAMP, and we never got those in mice. So I think maybe this is because in the non-Lewy body, since you have more availability of the toxic protein, maybe you have this like pumping the autophagy system. I don't know how, if you have an hypothesis on this. And the second is that well, I have it here. If you have some hypothesis of why, I mean, it's really fascinating for me that the non-Lewy body, they do nothing in mice, even if you leave them, because in the first study that you did with Mikel Vira, you left them for 17 months, I guess.
Yeah, correct.
They never got sick.
No.
And the monkeys, in the same span of time, they get the generation completely. So have you worked any hypothesis in the lab meetings or something about this?
So regarding your first question about the autophagy, so maybe, so as we inject no Lewy body, it's mainly form of oligomeric. So maybe the templating is much more efficient to create new oligomers, and maybe the perturbation is more related to cellular pathway, because for the no Lewy body, it's mainly related to substantia nigra and putamen, either compared to the Lewy body, which is much more cortical areas. So the pathology move to in other brain regions. So maybe as the aggregates were smaller at the time of injection, maybe the system created much more smaller oligomers, even after 2 years post-injection. And the dopaminergic cells were much more sensitive to smaller aggregates. So this is an hypothesis. And for an explanation between the mice and the monkeys, so maybe in mice, the no Lewy body are recognized as toxic species or not, or maybe the proteostasis system, so maybe the proteasome or the autophagy are much more able to clear small aggregates compared to monkeys. And the turnover is much more efficient maybe in mice compared to monkeys. Maybe it's a problem of protein or kinetics, or maybe the dopaminergic neurons can be also different between mice and monkeys. And as we inject also into the striatum, maybe the uptake between the mice and the monkeys can be different for the no-Lewy body. Maybe there is some receptors, or maybe the absence of receptor in mice compared in monkeys. So it was really a surprise for us. So now we try to understand why the no-Lewy body are much more toxic in monkeys compared to mice. So the first step is to characterize the structure of the no-Lewy body, and after to perform maybe some tests, maybe to come back to in vitro and to see between human cell lines and rodent cell line to see if there is some receptors, because some people identify some receptor such as LAX3. So maybe we have to investigate in those directions.
Okay, there is another question from Amanda in the chat. Is there any functional impact of the synuclein accumulation in the gut, digestive dysfunction?
It's a good question. So we know that so alpha-synuclein can be present in dopaminergic neurons, which are also present into the gut. But there is only some human studies, and it is quite difficult to count the dopaminergic neurons into the gut. So it seems that there is no neurodegeneration into the gut, even if there is some alpha-synuclein accumulation or alpha-synuclein aggregates. But it was mainly related to human studies. There is not much evidence in animal models, because it's quite complex to work with the enteric nervous system tissue. So we have to investigate, for example, we have some alpha-synuclein into neuron-specific enolase positive cells, and we have to see if there is some cell death. So we have to reanalyze our tissues. And the digestive dysfunction was a good point, because a reviewer asked for that. And as we didn't expect any neurodegeneration in the Lewy body injected monkeys into the gut, we were not able to follow those animals during the 2 years to see if there is some constipation symptoms or digestive dysfunction symptoms, because all the baboons were housed together in a huge aviary. So we were not able to answer to this question. But if we, when we will redo the experiments, we will take care to really follow those animals. So we will do a gut injection, but we will try to isolate those animals in individual cages and to check if there is some digestive dysfunction in those animals. So we were not able to answer to this question, but it was really a good question.
Okay. There are not any other questions. I thank Ben in the name of Achucarro for the talk.
Thank you.
Okay, I will stop recording.